Rain transforms a dry Ugandan pond into a nursery. In the mud, a seed pushes out a root, while nearby, a tadpole navigates the water. Welcome to Yaka Digital Network videos. Both of these organisms are experiencing growth, a strictly quantitative increase in physical size and mass, driven by cell division or mitosis. They are getting bigger. But getting bigger is only half the biological equation. They are also undergoing development, a qualitative increase in complexity, function, and specialization. Development is the process that takes a simple, singular structure, like that dormant seed or a spherical egg, and transforms it into a highly specialized adult form. The core mechanism driving this structural transformation is cellular differentiation. Every cell in these growing organisms contains the exact same DNA. But as they develop, specific genes are switched on or off, instructing identical cells to become distinctly different tissues, like a photosynthetic leaf or a highly sensitive nerve ending. This table formally compares the mechanisms of growth and development. Growth is easily measured by recording changes in height or weight, and is largely irreversible. Development, however, is measured observationally by tracking an organism's progression through distinct life stages to achieve an organized, specialized form. To understand how organisms actually achieve this functional complexity, we need to decode the distinct developmental blueprints of both plants and animals. Development is the ultimate biological partition, ensuring that identical microscopic building blocks are molded into entirely different survival machines. We start in the soil. A dormant seed is essentially paused in time. To begin developing, it requires 3 specific environmental cues: water, oxygen, and an optimal temperature. The very first step is imbibition. The dry seed aggressively absorbs surrounding water molecules, swelling significantly as its dormant internal enzymes finally activate. Once awakened, the seed must break through the soil surface to survive. Different plants employ different structural strategies to achieve this. The first strategy is epigeal germination. In a developing bean plant, the seed leaves, or cotyledons, are forcefully pushed above the soil surface line. The second strategy is hypogeal germination. In maize, the cotyledons remain safely hidden beneath the dirt, while only the shoot extends upward. Take a moment to test your recall. Which of these 2 distinct pathways pushes its cotyledons above the ground, maize or bean? If you identified the bean, demonstrating epigeal germination, you are correct. But regardless of the strategy used, the very first true structure to emerge from either seed is the radicle. The radicle develops downward, establishing the primary root to anchor the plant and immediately secure vital water resources. Following the root, the plumule emerges, moving in the exact opposite direction to secure sunlight for photosynthesis. Germination is a strict race against time. The seed must successfully differentiate its tissues into functional roots and shoots before its stored internal energy reserves run out. But underground, in complete darkness, how does a plant physically know which way is up toward the light and down toward gravity? This non-random directional development is known as positive geotropism in the roots and positive phototropism in the shoots. The plant relies on specific chemical hormones called auxins to translate these environmental cues into physical shapes. When sunlight hits one side of a stem tip, auxins migrate and densely gather on the unlit side. This high concentration causes shaded cells to elongate faster, mechanically forcing the stem to bend directly toward the sun. Structural development in plants is fundamentally a precise chemical response to environmental pressure. Animal development often involves even more drastic physical restructuring. In insects, this biological process is known as metamorphosis. This matrix details the 2 primary paths. On the left is incomplete metamorphosis, characterized by a 3-stage cycle, commonly seen in the local grasshopper. The middle stage, the nymph, looks structurally similar to a miniature adult, but it completely lacks wings and functional reproductive organs. To advance, the insect relies on ecdysone, the molting hormone. It dictates exactly when the nymph must shed its hard exoskeleton. Contrast this with complete metamorphosis on the right, mapped out in a 4-stage cycle, utilized by mosquitoes and butterflies. Complete metamorphosis establishes distinct life stages, assigning a completely separate biological job to each. The larval stage is exclusively a feeding machine. It operates with a simple tubular body plan designed strictly to consume nutrients and accumulate raw mass. By physically separating the feeding young from the flying reproductive adult, the organism effectively eliminates resource competition within its own species. The transition between that feeding larva and the flying adult happens in the third stage, the pupa. It is often labeled a resting phase, but that is entirely inaccurate. Inside the chrysalis, there is intense physiological violence. The existing larval tissues are systematically broken down and liquefied by powerful enzymes. Amidst this destruction, specialized cell clusters called imaginal discs survive. Fueled by nutrient-rich fluid, they multiply. These expanding discs differentiate, locking together to construct the complex features of the adult body: compound eyes, jointed legs, and wings. This level of physical overhaul is not limited to insects. Amphibians undergo a remarkably similar metamorphosis to transition from water to land. A tadpole swiftly morphs. The long tail is actively absorbed back into the body, and 4 strong limbs push outward for terrestrial movement. Internally, it develops lungs to process oxygen, and its digestive system shifts from processing a herbivorous diet to an actively carnivorous one. Whether encased in a pupa or swimming in a pond, reaching the adult form requires entirely dismantling the temporary structures of youth. Development is the highly regulated blueprint that dictates exactly how organisms arrange and specialize their cells to guarantee survival. This chart summarizes developmental hurdles. Plants, like germinating maize, must reach sunlight before exhausting seed reserves. Insects undergoing incomplete metamorphosis must grow while trapped inside rigid exoskeletons. Organisms executing complete metamorphosis must survive as vulnerable pupae while internal organs rebuild. This brings us directly to a standard UNEB examination application. Explain why a pupa stage is essential in complete metamorphosis, for 3 marks. To secure full marks, state that the pupa is the essential phase of intense physiological reorganization, where larval tissues are systematically broken down and destroyed. Then conclude that this breakdown provides the necessary resources to form mature adult structures, like wings and reproductive organs, strictly required for species propagation. Every complex biological form we see in nature is the direct result of a patient, highly organized progression through mandatory developmental stages. Thank you for watching. Watch more videos for all classes and get all content at www.yaka.cc.